Hi everyone, this is Linganagulu, your pharmacy student, and today we are going to discuss about the pharmacodynamics part 2. In pharmacodynamics part 2, we are going to discuss about the mechanism of drug action. In earlier video, we have discussed about the what is pharmacodynamics and the principles of drug action. So don't be late and let's get into the video. Pharmacodynamics. In simple terms, it can be referred as what drug does to the body. And today we are going to discuss about the mechanism of drug action. In previous video, we explained about the what is pharmacodynamics and the principles of drug action. If anyone has not seen that video, for your reference, I am providing the video link in the description. Going to the topic of mechanism of drug action. The mechanism of drug action describes how a drug interacts with biological system to produce its effects, and this helps in understanding in the crucial development for the development of therapeutic agents and predicting the clinical outcomes. And it also allows researchers to design drugs with specific therapeutic effects and predict the drug interaction and minimize the potential side effect. In simple terms, we can define it as the mechanism of drug action refers to the process by which a drug interacts with target molecules such as receptors, enzymes, or ion channels to produce a specific therapeutic agent. Going to the following steps of mechanism of drug action include drug binding and conformation changes, signal cascade, and response. So we will discuss in detail about these four following steps. At first, drug binding. It refers to the interaction between a drug and its target molecule, such as enzymes, receptors, and transport proteins. There are two types of drug binding. First one, reversible drug binding, and second one, irreversible drug binding. In reversible drug binding, most of the drug binds reversibly to their targets through non-covalent interactions, such as hydrogen bonds, ionic bonds, and hydrophobic bonds. In these types of binding allows for the dynamic regulation of drug activity. And second one is irreversible drug activity irreversible drug binding. In some drugs, form covalent bonds with their target molecule and that leads to the permanent inhibition. Example, certain enzymes inhibitors. And second one is conformation changes. It refers to the alteration in the three-dimensional structures of a molecule, particularly proteins that occurs in a response to a ligand binding or other stimuli. And it often determines how a drug interacts with targets, influencing their receptors for activation and enzyme activity. And in simple terms, we can say as the binding of drug causes conformational changes in the target molecule, which activates or inhibits its activity. And the conformation changes are two types. First one is allosteric modulation. In this, binding of a ligand to one site induces the changes and that affects the activity and at a different site. And second one is induced fit. In induced fit, upon binding, the drug induces a specific conformation changes in the enzyme receptors. Third one, signaling cascade. In pharmacology, a signaling cascade refers to the series of molecular events triggered by the binding of drug or ligand. It leads to the cellular response, and it is also known as signaling pathway or transaction pathway. The signaling cascade involves a sequence of molecular interaction and that ultimately leads to a specific cellular response. And fourth one is response. It refers to the biological effects resulting from interaction between a drug and its targets. There are two types of responses. One is pharmacological response. In this pharmacological response, there are two types. First one is agonist effects. Activation of receptors leads to physiological response. Example, pain relief from analgesic. And second one, antagonistic effects. In this, blockage of receptors inhibit physiological response. Example, antihistamines preventing the allergic symptoms. And second one, therapeutic response. In this, there are two types, desired effects and efficacy. In desired effect, the intended therapeutic outcomes such as lowering blood pressure with the antihypertensive. In efficacy, the maximum effect of a drug can be produced. Next, going to the fundamental mechanism of drug action. There are enzymes, ion channels, and transporters. So, at first, we go from the enzymes. Enzymes are biocatalysts, which are high molecular weight proteinous compounds, and it enhances the reaction which occurs in the body during various life processes. In pharmacodynamics, enzymes play a crucial role in determining how drug interact with the biological system. So, here type of enzyme, there are two types of enzymes. One is enzyme inhibition, second one is enzyme activation. In enzyme inhibition, drug can inhibit the enzyme activity by decreasing the production substrate of signaling molecule, and this can lead to therapeutic effect. Example, ACE inhibitors for hypertension. And in enzyme activation, some drugs can activate the enzyme by enhancing the metabolic pathway or cellular response. In example, certain products requires enzymatic conversion to become active and it perform the activity. And next we are go for the ion channels. Ion channels are the integral membrane protein that facilitate the movement of ion across cellular membrane, and they play a very crucial role in the physiological process. And ion channels are the target for many drugs which modulate their activity to produce therapeutic effect. And there are different types of ion channels are there. They are mainly four types of ion channels are there. First one, voltage gated ion channel. Second one, ligand gated ion channel. Third one, mechanosensitive channels. And fourth one, leak channels. So first one, voltage gated ion channel. So it performs the activity by open or closing response to change in the membrane potential. Examples includes sodium channels and potassium channels. And second one, ligand gated ion channels. So it performs the activity open in response to the binding of specific ligand, and examples are neurotransmitters like acetylcholine. In third one, mechanosensitive channels. So it responds to only mechanical stress of the activity. Example, stretch or pressure. Fourth one, leak channel. So it performs the activity by constantly opening and allowing the ions to flow freely and maintaining the rest membrane potential. So the function of ion channels includes it regulates the ion flux and the membrane potential during the activity process, and it controls the muscle contraction, nerve impulse transmission, and secretion. Transporters. The transporters are the specialized protein that facilitate the movement of drugs, nutrients, and other molecules. The transporter also play a crucial role in absorption, distribution, metabolism, and excretion of pharmaceuticals. There are two types of transporters are present. First one is solute carrier transport, and second one is ATP binding cassette transporters. In solute carrier transport, it mediates the transport of small molecules including ions, sugars, and amino acids. Examples are glutamate transporters. And second one, ATP binding cassette transporter. It uses the ATP to transport a wide range of substances, often in drug efflux and impacting drug resistance. Example, P-glycoprotein. So guys, this is the topic for today, and we can discuss about the receptor pharmacology in next session. I hope everyone liked this video. So please like, share, and subscribe to Pharmacy Student.